Good morning, my dear students. This video lecture is about isolation of microorganism. Myself, Dr. Shibu, Assistant Professor in the Department of Biotechnology, Safi Institute. Coming to the isolation of microorganism, you have already know what is mean by microorganism, and this lecture is about how to isolate the microorganism as a pure culture. So microorganisms are generally found in air, soil, and in water as a mixed population. Even the disease part of the plant and animals contain a great number of microorganisms, which may be differed from microorganism from other environment. One microorganism present in on such environment is different from the other environment. To study the specific role played by a microorganism in its environment, one must isolate the same in pure culture. So if you want to study about the microorganism, you need to isolate the microorganism in pure form. So again I will tell you, in pure form you cannot able to give a contaminated microorganism for the study. So two major step of obtaining a pure culture as have been described here. First, the culture has to be diluted until the various individual microorganisms are separated far apart on agar surface. That after the incubation, they form visible colonies isolated from the colonies of the microorganisms, which means we need to culture the microorganism and we differentiate from the plates needed, diluted the colonies. We need to reduce the crowded of the colony with the help of dilution, serial dilution, and we need to grow the microorganism the plate by visibly seeing the colonies in the plate. We can confirm the growth of microorganisms. Second, an isolated colony has to be aseptically picked up from the isolated plate. Then what we need to do is we need to isolate the colony, separated the colony from the other colonies. We need to pick up with the help of the loops. Okay. So what is the difference between pure and mixed culture is, here you can see that the first plate, that is a plate, it is mentioned pure originate from one bacterial strain. All the colonies are looked same there. You can see that it is maybe in orange color, it is it's a pure one. All the colonies are same. But in the second plate you can see that there is a shape and the size of the colonies are different, which maybe leads to a different colonies or mixture of colonies. That's what it is, a mixed colony, or we can commonly say that it may be contaminated with other organisms. So that is the difference between pure and mixed culture. So point to be taken into your account is, during the inoculation, the inoculation loop has to be sterilized before each inoculation of colonies from the agar plate. Once you are inoculating the culture, you need to sterilize your loop with the help of the heating, with the help of their flame. It is present inside the lab. Okay. So every time the loop is sterilized by heat and it is cooled before the inoculation to the next colony. Once you are streaking or spreading, you need to heat or sterilize your loop or your L-rod. There are several methods of isolating the pure cultures of bacteria, like streak plate method, pour plate method, spread plate method. Then you have having a serial dilution. Streak plate, pour plate, and spread plate method are commonly used to isolate the pure culture. So what is the important of pure culture are, it is once it is purified, the isolated species can then be cultivated with the knowledge that only the desired microorganism is grown. Okay, that is the purified colonies mean only the desired colonies are there. Then a pure culture can be correctly identified for accurate studying and testing and diagnosing in the clinical environment. So a pure culture can be correctly identified for the accurate study. Then only you can able to know that which microorganism you are studying and which microorganism you are going to test. So that is the importance of pure culture. Then testing or experimenting with a pure culture ensure that the same result can be achieved how many times you are doing the test repeatedly. Whenever we are repeating the test also, you should have to get the same result for a same pure culture. Okay, so the pure culture, the spontaneous mutation rate is very low, and they are mentioned about it as the clone is 99.9% identical. Then common methods for isolating the pure culture. The process of screening a pure culture by separating one type of microorganism from the mixture is called as a isolation. From the mixture of colony, if you are isolating a particular colony or particular strain, that is called as an isolation. Okay. So certain common method, as I earlier said, streak plate method is there, or pour plate method is there. Pour plate means loop dilution method or serial dilution technique. Then spread plate method is there. Then micro manipulator method is there. Then roll tube method is there. Okay, micro manipulator. This first three are very common: streak plate, pour plate, and spread plate. We already know about the things. Then micro manipulator is a type of an instrument to pick the colony. Okay, so to pick a particular colony, we'll use the micro manipulator apparatus or an instrument. Then roll tube method, just rolling the tube. We need to this is commonly used for anaerobic type of microorganism. So first is a streak plate method or a streaking method. So in this method, you maybe already know that we are having a loop. Okay, so the loop will be sterilized. You can the diagram, it is the picture is mentioning about the loop. The loop should be heated, and with the help of the loop, you should have to take a loop full of colonies. Okay, colony, and it should be streaked on the media. So different type of streak, maybe aware about quadrant streak, symbol streak, T streak, etc. So of that, quadrant streak is good for isolating the pure culture. Okay, so there are certain steps which will be mentioned for isolating the things, and the diagram also mentioning about the quadrant stick. Here you can see that quadrant stick to reduce the colony count, or you need to get an isolated pure colony. So it is mentioning first you should have to hold the broth culture containing in the tubes in your left hand and shake it. Then the second step is sterilize the wire loop of inoculation needle in the burner. Then remove the cotton block of the broth and broth culture tube by your little finger of the right hand. Then flame the mouth with, of the test tube. Then insert the loop or insert the wire loop into the flask or the tube. Then the thin film in the loop is streaked in either a zigzag manner by removing the loop backwards and forward firmly. Maybe already aware about doing quadrant streak. Then incubate the plate in an incubator at the required temperature, and growth of the bacteria will be visible on the streak mark, and when you are culturing or put it in an incubator for overnight. Next is a pour plate method. This pour plate method is a bacterial culture and the liquid agar medium are mixed together. Here we are not spreading or streaking above the agar media. Here we prepare the agar solidified, that is a liquid agar media, and mix the culture. After mixing the medium containing the culture poured into the sterile petri dish. Okay, and allow it to solidify and incubate. And here you can see the colony should be appeared on the surface. Okay, it should be embedded into the surface. Okay. So this is the diagram what is mentioning. So maybe you already aware about this doing the pour plate, mixing of the media with the culture, and shake it and pour it on the plate. Okay, then after overnight you can see the colonies. Certain disadvantages here that you mentioned about pour plate is, so the most probably the microorganism trapped beneath the surface of the media when it's solidified. When we are mixing the media with the culture, when we are pour it on the plate, there is a chance of the microorganism to settle down at the plate. That is one of the disadvantages. We cannot able to isolate properly. Then the method is tedious and time consuming. Then some of the microorganisms were subjected to heat shock because in the case of spread plate or streak plate, after the solidification and cool, we are spreading or streaking the microorganism above the surface of the plate. But here it is not like that. Before solidifying at the 45 degrees Celsius, we need to mix the microorganism with the media, so that some of the microorganism cannot able to tolerate the heat shock, so it will be died at the 45 degrees Celsius. So this method is unsuitable for isolation of psychrophile bacteria. Psychrophile bacteria means the bacteria which is living on a extreme cold condition bacteria cannot able to tolerate a shock. Okay, so that is a disadvantage of pour plate method. So procedure just mentioning about the pour plate method. So you already know about this spread and pour plate method. So for the first diagram, it is mentioning about spread plate method. We need to prepare a plate containing the required media, and around 0.1 ml or less amount of solution which containing the bacteria. Suspension solution should be put it on the plate, pip it out and put it on the agar surface, and within the L rod we need to streak it. Okay, so streak it properly, we need to spread it properly, and we need to incubate it, and above this, after incubation, you can see the colonies present above the surface. And pour plate is both the things, the media and the culture is mixed together, and you can pour it on the plate, so that you can see the colonies in the plate after the incubation. That's what it is mentioned here. Then serial dilution. So you may be aware about serial dilution. This is a method commonly used to obtain pure culture of those microorganisms that have not been successfully cultivated on a solid media or growing on a liquid. Okay, there is an to reduce the colony count. Usually you need to perform the serial dilution. We are preparing a 7 tubes with 9 ml and we are diluting the content of microorganism. Okay, serial dilution. So this diagram which is mentioning about the serial dilution, you can see that if you need to take 1 ml of the media and mix it with 9 ml of fresh sterile liquids. Okay, so we would have 100 microorganism in 10 ml or 10 microorganism per 1 ml. Okay, so like that we need to serially diluted that sample. Okay, so this is called a serial dilution. Okay, after serial dilution, so most probably for the bacteria 10 power 5 to 10 power 7, that is suitable for taking the bacteria. That plate is taken into count, or in the case of fungus it is 10 power minus 3 to minus 5. Then the final one is spread plate method. So this plate method, as we earlier mentioned about, with the help of LRAD, we need to spread the cultures. Okay, so we need to spread the culture above the surface of the plate, that we already mentioned in the diagram. So that here it is mentioning about how to perform the experiment. So here you can see the diluted bacterial sample should be taken 0.1 ml and spread it on a nutrient agar, and then you can see the isolated colonies above the surface of the plate. Then the advantages of the spread plate method is very simple, and the colony will be absorbed above the surface. That's what it mentioned, and microorganisms are not exposed to high temperature. That is the advantages of this spread plate. Okay. Next is micromanipulator method. As I earlier said, it's an instrument which is used to pick the colony. So it is attached with the microscope so that we can pick a single colony from the plate. For example, if the plate is having 100 or 150 colonies, we cannot able to identify which colony we need to take. Okay, so in order this micromanipulator is connected to the microscope, so that within microscopic condition we can pick the perfect colony. So it is having advantages and disadvantages also. Advantages is we can reasonably sure that culture can from a single cell, and one can obtain a strain with the species. So from the mixed population you can pick the single strain. Okay, here in the disadvantages, this equipment is expensive, and since it is expensive, it require a tedious work and need a skilled operator. That is a disadvantage. So this is the instrument. You can see that micro manipulator connected with the microscope. Okay, so that's all for today. So hope you will understand the thing. Okay, so what we discussed today is about isolation of microorganism with the help of spread plate method, pour plate method, and stick plate method. Okay, so the remaining we'll see in the next class. Okay, thank you.